Hello Biology 230 at Tuskegee University. This is Dr. Griffin with Module 11, focusing on the molecular basis of inheritance. Module 11 discusses the major concepts of Chapter 16 in your textbook, and in this video I will give you a brief introduction to how DNA was discovered to be genetic material. We have to thank Dr. Frederick Griffith for doing his famous experiment, which is just known as the Griffith Experiment. So I'm not talking to you today about this experiment just because I like his last name. But indeed, it was one of the earliest, if not the earliest, experiment to really show the transformative power of deoxyribose nucleic acid. In this experiment, Dr. Griffith used living S cells, and these living S cells were pathogenic bacteria cells. Okay, they were bacterial cells that were pathogenic, meaning that when you injected these pathogenic bacteria into mice, the mouse would indeed die. Now, in another set of mice, they were a little lucky because he injected living R cells, and these are the same strain of bacteria except for they are non-pathogenic, which means that these bacteria are indeed alive, but they do not cause disease in the mice, and actually the mouse remained healthy and remained alive. The next step was then that he actually killed bacteria, so he used heat to kill the S cells and injected these killed S cells into mice, and they remain healthy. Remember, these are pathogenic. The S cells are pathogenic, so they should be causing disease. But since he killed them with high heat, high temperature, the mice were indeed remained healthy. Now here's the cool part. He mixed heat-killed S cells. These are the pathogenic ones. They're supposed to be killing the mice, but yet he killed them, so therefore these bacteria, these pathogenic bacteria, are not alive. But he mixed them with living R cells, the non-pathogenic bacteria. What did he discover? He discovered that mice injected with this mixture, all of them died. And not only that, when he took samples from the dead mice, he discovered living S cells. Well, how could that be? How could he obtain living S cells from mice that were injected with dead S cells? How did these S cells come back to life? Well, they did not. They transformed these living R cells into living S cells. And that stuff that was able to transform the living non-pathogenic cells into living pathogenic cells, pathogenic bacteria, is DNA. I want you to think a minute about how this experiment proves that last statement that I just made. Because you can say, well, maybe it was the protein, or maybe it was the cell wall. How does this experiment conclude decisively that it was not the cell wall of the bacteria that transformed R cells into S cells, but it was something indeed inside the cell? It was something indeed, something intact, something internal in the cell, and we now know it as DNA, our transformative agent. This experiment showed that DNA is the genetic material. It is that material that is heritable. It's the stuff that programs cells and gives cells their identities. This is the end of video 1 out of 4, so you have 3 more videos to watch for Module 11, and not just watch, but also learn from them. This is Dr. Griffin, and I'm signing out today. See you next time.